Hello, we shall discuss on the biological classification. The earliest attempt to provide classification was done by a person named Aristotle. He distinguished plant and animal on simple identification. He classified the plants into herbs, shrubs, and trees using simple morphological characters and divide the animals into two groups. One is two groups on the basis of the color of the blood. That is, those animals that have red color blood and those animals that do not have red color blood. So he classified animals on the basis of the color of blood. In the meantime, a scientist named Carolus Linnaeus gave two kingdom system of classification. So under this two kingdom system of classification, the two kingdom were the kingdom Plantae and the kingdom Animalia. So Carolus Linnaeus, he classified all the animals and grouped under this kingdom Plantae, and all the animals were classified and put under kingdom Animalia. However, this two kingdom system of classification was proved to be inadequate because of the following reason. The first one is that the two kingdom system of classification did not distinguish between the eukaryotes and prokaryotes. Eukaryotes are those organisms having well-developed nucleus and nuclear membrane in the cell. Example is plant cell. The prokaryotes are those which they do not have well-developed nucleus and nuclear membrane. Example is you can take bacteria. So the two system of classification did not distinguish between these prokaryotes and eukaryotes. The first reason. The second reason is that it also did not distinguish between the unicellular and multicellular organisms. Unicellular are those organisms that have only one cell. Multicellular are those organisms which have many cells. That means the body is made up of many cells. The example of unicellular, you can take bacteria or amoeba, and multicellular is plant animals. So distinguishing between unicellular and multicellular is also not done. So this is the second reason. The third reason is that it did not distinguish between the photosynthetic and non-photosynthetic organisms. Photosynthetic are those organisms which can prepare their own food by the process of photosynthesis, and non-photosynthetic organisms are those organisms which cannot prepare their own food. That means they depend on other organisms for their food. So distinguishing between photosynthetic and non-photosynthetic is also not done by this two system of classification. So this is the third reason, and these are some of the reasons why two system of classification provided by Carolus Linnaeus was found to be inadequate. Now we shall discuss on the five kingdom system of classification. Five kingdom system of classification was proposed by a scientist named R.H. Whittaker in the year 1969. So the five kingdom system of classifications include following kingdom. The first kingdom is Kingdom-Monera, the second is Kingdom Protista, the third is Kingdom Fungi, the fourth is Kingdom Plantae, and the fifth is Kingdom Animalia. The main criteria that is being used by this scientist for doing five kingdom system of classifications are: he included cell structure, body organizations, mode of nutrition and reproduction, etc. Now we shall discuss on the five kingdom system of classification. The first one, let's discuss on the Kingdom-Monera. Under this kingdom, bacteria are the sole members. And these bacteria are found to occur in almost everywhere in the planet. They live in extreme habitats such as hot springs, desert, snow, and deep oceans where very few life forms can survive. Some of the bacteria also occur as parasites living on some other organisms' body. Bacteria are grouped under four categories on the basis of their shape. The first group of bacteria is the coccus. The plural form of coccus is cocci. So those bacteria which occur in spiral shaped are called as coccus. The second is bacillus, and the plural form of bacillus is bacilli. So those bacteria which occur in rod shaped are called as bacillus. The third group of bacteria is the vibrium. Plural is vibrio. So those bacteria which occur in coma shaped are called as the vibrium or the vibrio. And the fourth group of bacteria is the spirillum. The plural form is spirilla. So those bacteria which occur in the form of spiral are called as spirillum. Now let's discuss on the mode of nutrition of this bacteria. Bacteria can be both autotrophic and heterotrophic. Autotrophic are those bacteria that can prepare their own food. That means they can synthesize their own food. These autotrophic or autotroph bacteria are grouped into two. There is photosynthetic autotroph and second is chemosynthetic autotroph. Some bacteria like cyanobacteria, also called as blue-green algae, have chlorophyll that is very similar to that of the green plants, and through this chlorophyll they can synthesize their own food in the presence of light. This bacteria also possess a specialized cell called as the heterocysts, which are characterized by a thick wall. So these heterocysts are found in the cell of the bacteria, and this heterocysts, the importance of this heterocysts is that this heterocysts, the presence of this heterocysts in the bacteria help to fix atmospheric nitrogen into usable form. So some of the bacteria that fix atmospheric nitrogen by the presence of heterocysts are nostoc, Enebena, etc. The second autotrophic group of bacteria is the chemosynthetic bacteria. Chemosynthetic, these are those bacteria that can oxidize various inorganic substances into usable forms. They cannot convert, they convert the inorganic compounds into usable compounds like nitrates, nitrites, and ammonia, and use the energy that is being released while converting this inorganic into a usable form. So this chemosynthetic bacteria plays an important role in nutrient cyclings like nitrogen cycle, phosphorus cycle, and even sulfur cycle, etc. Now we shall discuss on the types of bacteria. The first types of bacteria under this Kingdom-Monera. So the first bacteria is the Archaebacteria. Archaebacteria are special type of bacteria living in some of the most harsh environmental conditions or most harsh habitats like extreme salty areas, hot springs, and marshy areas. So those bacteria which are found in extreme salty areas are called as halophils. Those bacteria which are found in the hot springs are called as thermoacidophils, and those bacteria which are found in marshy areas are called as methanogens. So Archaebacteria differ from other bacteria in having different cell wall structure which helps them to survive in extreme environmental conditions. Now methanogens. So these methanogen bacteria are also found in the guts of several ruminants animals like cows and buffaloes, and these methanogen bacteria help in the production of biogas from the dungs of these animals. Now eubacteria, we shall discuss on the second type of bacteria, that is eubacteria. Eubacteria are also called as the true bacteria, and this group of bacteria are characterized by the presence of rigid cell wall and flagella. So these bacteria can move because of the presence of flagella. The mode of nutrition of this group of bacteria can be both autotrophic and heterotrophic. Now let's discuss on the heterotrophic type of bacteria. We have discussed about the autotrophic type of bacteria, that is autotrophic consisting of autotrophic can be both photosynthetic and chemosynthetic. Now we shall discuss on the heterotrophic bacteria. Heterotrophic bacteria are those group of bacteria that depends on dead organic matters. They are the most abundant group of bacteria in nature and are mostly decomposers. That means they can decompose the organic matters and release them back into the environment. This group of bacteria are also important in processing of foods in industries and for production of antibiotics. Bacteria like lactobacillus help in the conversion of milk into curd, and bacteria such as bacillus help in the production of antibiotics. Some bacteria like rhizobiums also occur in the leguminous plant and form a symbiotic association. So in this association, their bacteria help in the fixation of atmospheric nitrogen into usable form. Now let's discuss on the reproduction of bacteria. Bacteria reproduces mainly by process of fission. Under this method, a single cell of a bacteria divides to give rise to two bacterial cells that can grow and develop into independent bacteria. However, under unfavorable condition, bacteria reproduces by forming spores. So the formation of spores for reproduction is called as sporulation. So bacteria reproduces asexually by the formation of spores.